This conference will now be recorded.
Hello, good afternoon, everyone. Welcome to the Barshop seminar. I'm going to go ahead and get us started because we have 3 of our Biology of Aging postdoctoral fellows presenting today, and I want to make sure to give each of them enough time. And so I am going to welcome Dr. Atrian for the first presentation. Maybe.
Hello. Yes. Hi. Hi, everyone, and good afternoon. Dr. Frost, she's trying to join, but apparently it doesn't let her to join.
Yeah, so we just said for anyone that's getting something like that, we just sent out a corrected link. So the one that we sent earlier wasn't working. So if she checks her email, I'll email it to her again, but she may need to check if somebody has a fast way to tell her.
Okay, so should I start?
If you want to share your screen.
Yeah, that's what I'm trying to do, but
I've made you the presenter.
okay.
Do you have the option at the bottom of your screen to share?
Actually, it doesn't let me to share my screen.
It looked like something happened, so now try. Still not giving you the option?
No.
What does it say?
It says she's the presenter. I don't know if you've enabled screen sharing specifically.
I have no idea.
If she's the presenter, she would have the ability to share her screen. There's no other mechanism with GoToMeeting for a person to share their screen. So it should be on the bottom, the screen.
Yes, I can see that, but it's
Is there a little X through it?
Is it possible you're signed in twice? I see 2 of you in the group.
Yeah. Let me
Maybe
connect.
Yeah, now make sure the one that's remaining is the one that's the presenter.
Now I don't see her to make her the presenter. Is she on here still? Oh, there she is. I see. Wait, now there's 2. I don't know which one.
There's still 2 copies of her.
I don't know which one to make presenter. Maybe that's the problem.
Let
me try this one. Now, it could be the wrong one was presenter. See if it works now.
There's only one now, so yeah.
Okay. Thank you.
Oh, I'm sorry, but it is asking for it. It doesn't let me. Is it possible someone else to start and I start after them? Because I think I need to fix this. I don't know.
Of course. Cody, are you comfortable starting now?
Yep.
Okay, good, because I have to find you too. All right, let's hope you have permission. All right. Thank you.
Seems to be working. Okay. Yeah.
Yep, I can see you good.
Okay, cool. Okay. my name's Cody. I'm a biochemist, so I'm going to present data a little bit different than typically comes up in these seminars. Thank you for inviting me to do this also. And so I'm a postdoc in Patrick Sung's lab, and we are interested in DNA repair mechanisms, and specifically double-strand break repair. And the main pathway for double-strand break repair that we are interested in is homologous recombination. And so I'm just going to quickly go over this really oversimplified diagram of kind of what happens when cells encounter a double-strand break, which can come from DNA lesions or replication fork stalls. They can be programmed for sister chromatid exchange, things like that. But basically, when cells encounter a double-strand break, they have mechanisms that bring in nucleases and other cell cycle cascade signaling, helicases that come and resect the DNA to leave long 3' tails that are subsequently loaded by kind of the central factor in homologous recombination called Rad51. Rad51 is involved in the homology search. So this strand searches for homology from the undamaged sister chromatid and uses that as a template for repair synthesis and then re-annealing basically of the two strands back together in a manner that's considered to be error free as compared to other pathways that can be more deleterious, like microhomology or strand annealing type pathways that kind of don't they still resect but aren't able to permit repair synthesis and then just kind of put things back together, or non-homologous end joining, which puts broken ends straight back together, which is considered to be more error prone. And so one of the, I guess, classes of proteins we're interested in is BRCA1 and its and it forms a homodimer with a protein called BARD1. It interacts with other proteins that are mutated in cancers and premature aging diseases. And so what I really want to focus in on is this step of homologous recombination right here, which is the strand invasion step. So a lot of effort and resources from the cells are dedicated to making sure this happens right. So when cells resect DNA, the first thing that encounters the single-strand DNA, which needs to be protected from further nucleolytic degradation, is RPA, because it binds DNA so tightly. There's tons of it in the cell. So that needs to be removed and loaded on with Rad51, and then which is mostly thought of to be promoted by BRCA2. There are other accessory factors that we're starting to see promote that step as well. And then once that Rad51 is loaded, BRCA1 and BARD1 promote the strand invasion step. So they interact with this Rad51 filament, help in the synaptic complex formation where the Rad51 searches for homologous DNA on the sister chromatid, and then repair synthesis occurs. And then there's even more downstream steps that are involved in kind of refilling and getting everything back to normal. There's crossovers and non-crossovers and things I'm not going to get into. But the main thing I want to bring up is we know BRCA1 BARD1 is important for this step, and despite there being thousands of papers on BRCA1 BARD1, their role in different steps of this repair process, we really don't know that much about the actual like intimate details of how these proteins function in this pathway. And so part of the reason is they're big proteins and they have tons of interacting partners. So BRCA1 BARD1 are kind of a constitutive heterodimer that interact through this ring domain mostly, and then it's been shown partially through the BRCT repeats on the C-terminus. And both proteins have Rad51 bindings ability, DNA binding. They interact with other repair factors. I mentioned POLB2, which also bridges BRCA1 to BRCA2, and POLB2 comes with its own suite of DNA binding and Rad51 interacting domains. So basically, we have this giant pool of protein interactors. We don't know what any of these different interfaces do. And so I just put these dotted lines for the Rad51 and DNA binding interaction interfaces because we don't actually know where those are in the protein, and we also don't know what they contribute to its activity, especially in the context of homologous recombination. So the two major questions that I look at when I work on this project is, how do these HDR components work together to promote DNA repair? And specifically, I'm just going to focus on kind of that strand invasion step today. So how does BRCA1 and BARD1 interact with POLB2 and BRCA2 and other Rad51 like proteins? And then within those proteins and within those complexes, how do discrete domains, the DNA binding domain in BRCA1, the DNA binding domain in BARD1, why do both have a DNA binding domain? What do they contribute to different activities? And so that's led us to kind of this two-pronged approach in tackling these questions. So the first one is to purify complexes of these proteins and show how they promote these activities biochemically together. So BRCA1 promotes D-loop activity, the strand invasion step of Rad51, but so does POLB2. Do they work synergistically? It's an easy prediction that they probably would, but we don't actually know. BRCA1's mostly thought to just bring POLB2 to the site of damage, which then brings in BRCA2. But I think what we're starting to see is it's probably a lot more complex than that. So we're trying to figure out how these different pieces work together. And through that, we're also doing cryo-EM, or we're collaborating with people to do cryo-EM of some of these larger complexes. Newer technologies for cryo-EM have made it possible to look at big protein complexes like this and get some structural information, which helps us kind of understand how these proteins are working together. And then when we have these complexes, what happens if you knock out the DNA binding domain of one of them, or the Rad51 binding domain, or the BRCA1 POLB2 interaction interface? Do they still work together or not? Those are the things we don't know. And then on the other side of it, in a more, I guess, small scale, intimate look at these proteins, we're taking those putative domains that I showed before, the DNA binding, the Rad51 interacting domains, and really trying to narrow down specifically which regions of the proteins are important for those, and then even beyond that, which residues are important for those interactions. And those smaller fragments give us options for like NMR. So we're working with David Libich to do some NMR to get some structural information on these interfaces. And then from there, we can design mutants that disrupt just DNA binding or just Rad51 interaction, and then take those and add them to our biochemistry assays. We can look for changes in phenotypes, drug sensitivities, all that stuff in vivo. And we can use these fragments to start to screen for chemical inhibitors of various interactions. So as a biochemist, a lot of my time is spent purifying protein, and these are about as hard as it gets to when it comes to purifying protein. These are big. I'm bringing multiple pieces together. So this is relatively challenging part of the project, is just getting the material to do these things. And so I've been spending most of my time optimizing these. And so we typically use insect cells. They're another eukaryotic system. They express these proteins okay, for the most part. And then we do a couple step affinity purification, and then size exclusion chromatography is just another purification step that helps separate the protein and make sure it's not aggregated, so it's nice monodispersed, happy behaving proteins to use in our biochemical assays. And so I've got examples here: BRCA1 BARD1, BRCA1 BARD1 with POLB2, and POLB2's interaction, or an interactor, which we don't know what MRG15 is doing at all. But I basically have got these materials gathered up, and I'm spending most of my time applying them to assays now. And so right now, everything is building this tool set to kind of put together to start answering those questions that I wanted to ask before. And just to give you an example of one of those ways we do that, there's this D-loop assay that we work on a lot, which mimics the strand invasion step of RAD51. So what we do is we take a Cy5, this is just a fluorescent labeled DNA, incubate it with RAD51, and then whatever factor we're interested in, BRCA1, POLB2, and then that creates this RAD51 filament, and then we incubate it with the supercoiled plasmid that has homologous sequence to this DNA, and it forms D-loop. And then we run those on a gel to look for changes in migration, which indicates product formation. So my recent BRCA1 BARD1 preps, this took some troubleshooting to get to this point, and there's still some optimization that needs to be done here. But as has been published before, BRCA1 BARD1 has D-loop activity, and my protein binds DNA. So this is an EMSA. So this is different DNA substrates all mixed together. And then we titrate in protein, and we look for shifts in the DNA to get a sense of what substrates are preferentially bound by our protein. So all of this is just to say that I've got active protein and kind of this, what I think of as a platform to really build on. So now the next step is to take all these pieces and start to add more to them, start adding in POLB2 and other things that I don't have time to talk about today, but we've got a few applications that we're primed and ready to test now. So I just want to shift gears to the other aspect really quick. So one of the ways we look at different interaction interfaces is we've purified different fragments of BRCA1 and BARD1 or whatever we're interested in, and we test them for interactions with DNA using the EMSA like I showed before, or we just do pull downs. So we'd narrowed down previously that these are kind of putative sites for DNA binding and RAD51 binding here, but we don't know exactly what residues are important. And the smaller we get, the closer we get to finding what residues are important, and it makes structural biology via NMR easier. So we're trying to narrow down specifically where the binding works. And so a lot of this work has been done by an MD PhD student in our lab, Angie Jasper. And so she started off by taking this largest portion that we knew bound DNA, 467 to 696, which just refers to the residues that are in this fragment. And she started chipping away at trimming down the C-terminal or the N-terminal portion of the protein, and found that all of these different substrates that we're interested in were bound with basically the same affinity. And so we thought maybe we had started to trim it down, and we want to start trimming it down even further. So she looked at the other side. She trimmed off the C-terminus and then found that bound DNA. So she trimmed off both the N-terminus and the C-terminus, thinking that this was probably our new smallest fragment that bound DNA. But it's obviously, as most things are, was more complicated than that. So what happened was she was testing the binding of these BRCA1 fragments to forked DNA. And she saw that the largest fragment and then the trimming of the N-terminus or the C-terminus all bound DNA the same. So when she went to look at the smallest version of that, this has both the N-terminus and the C-terminus trimmed off, she saw that DNA binding was eliminated. And so that left us with this kind of puzzle here. So this bound protein, this bound protein. So we thought maybe this blue section was where DNA binding actually was, but when we purified and tested that for binding, we saw no binding. So what we did is we took increasing amounts of salt. So these are electrostatic interactions for the most part with DNA. So by increasing the salt, we can basically be more selective for good DNA binding. So all the fragments bound DNA at our standard salt, 90 millimolar KCl, more or less the same. But once we start increasing the amount of salt, we see bigger differences between the binding. And so where we're kind of heading with this is we think that maybe both regions actually have some connection to DNA binding. So whether or not they're two independent binding domains or in 3D space, they're actually next to each other. It'll take structural biology, NMR structures to really figure that out and tease that out. The other alternative is that both sides bind DNA, but maybe one side has preference for one substrate or another. So those are the types of questions we're going to follow up on. And I don't want to get spend too much time on this because things are winding down, but we're also doing the same approach with BARD1, but I just wanted to show this because this is kind of a different way to look at, this is actually making mutations. So we knew that this 124 to 270 region of BARD1 bound DNA based on previous fragments. And so we can take positive residues that we see, these lysines here, especially ones that are clustered, are obvious choices for disrupting DNA binding. And so as kind of we predicted, if you mutate these residues from positive charge to negative charge, you can eliminate DNA binding. So we don't know which of these Ks are the real important ones yet, so we need to test those. And the tricky part here is the BARD1 DNA binding domain seems to overlap with the RAD51 binding domain. So this 8K mutant also knocks out RAD51 interaction. So we're not getting, we need to be able to separate the two functions so we can really understand what's happening in our other biochemical assays and in cells. So just to wrap up, I'm working on purifying more complexes, more combination of complexes, and applying them to the D-loop assay. And then once that's really cemented and running smoothly, then we can start to expand from there. And I think we'll start to see some really exciting stuff coming out of that in the coming year. Then we're looking for point mutants that disrupt interactions. And again, we can apply these to our various assays. And we're also working with others. And the biochemistry and structural biology department is hiring more structural minded people. So I think we're going to have a shot at getting some structural information from these, which is really useful. And it all kind of ties back into it together and to paint the whole picture of how these proteins are working together. So I don't have the whole full sung lab here. I just want to say thanks to a few people, mostly Angie, who did a lot of the DNA binding assays for the fragments, and then thank you for the T32 Biology of Aging training grant, which helps support my research. And I can take questions now. I see there's one here in the chat. Any evidence these somatic mutations are associated with age-related increase in cancer risk? So I don't know if they're related to age-related increase in cancer risk directly. I think the jury's still out on that. The connection between the BRCA1, BRCA2, POLB2 to aging isn't really strong at the moment, but we have evidence in the lab that proteins like the RET-Q helicases that are directly involved in aging diseases are involved in this process too, but that hasn't really been defined. So I don't know of the direct connection to age and cancer risk when it comes to these proteins. These and then so are the data presented in cell cultures and or cell-free assays. So this is all biochemistry in vitro, just labeled DNAs and purified protein. So I think that's it for the chat questions. I don't know if anyone has anything else.
Thank you so much, Cody. I think we're still having some technical issues we're trying to go around. And so potentially, Julia, could you go next?
Yeah, I can make her directly.
That would be lovely.
Okay. All right, can everybody see my screen?
Yes, I can see it.
Okay, let me get my pointer. Okay. All right, well, thanks for having me talk today. I'm Julia. I actually work over at Texas Biomed in Joanne Turner's lab, and I'm going to be shifting gears and talking about the aging immune system. And so specifically, I'm going to be talking about some work that I've been doing in the lab looking at how increased age impacts vaccine-induced tissue immunity. So as we all know, and we've seen recently with the importance of the immune response in COVID, that with increased age, there are changes in the immune system. And so one of the major results of this is that with increased age, we have decreased immune responses to vaccination and challenge. And so in elderly individuals, so those over the age of 65, we know that this population is both less responsive to vaccines, so they generate less of a protective immune response, and they also are more susceptible to infection. And so some of the major threats that we normally think of are flu and pneumonia. And then, of course, recently with SARS-CoV-2, we find that in the elderly population, worldwide, about 80% of hospitalizations and 3/4 of deaths are in those over the age of 65. And so SARS-CoV-2 has really highlighted how susceptible this population is to infection. So just a little bit of background to talk about the immune response when we receive a vaccine. So we can think about the immune response, either antibodies or T-cell mediated immunity, as a function of time. So say you go to the clinic, you receive a vaccine, you are going to generate a primary immune response. This is going to happen either days to weeks, depending on the vaccine. And the hope is that then when you come back into contact with this pathogen that you've been vaccinated against, that you're going to generate a faster and a larger and more robust secondary immune response. And so this will really help you to protect yourself against that secondary challenge. Unfortunately, we know in those that are older, the primary and secondary responses are delayed and decreased. And so this is adding to this increased susceptibility. So in our lab, we study aging in the context of the disease tuberculosis, which is caused primarily by the respiratory pathogen, a bacteria, Mycobacterium tuberculosis. And in older individuals, we know that they are both more susceptible to developing disease and then succumbing to the disease. And so some work from our lab that I'm going to show here has looked at T-cell responses in the peripheral blood that we've obtained from human donors. And so to orient you, this on the x-axis, these are our different groups. And so the adult and elderly controls are going to be TB naive. Then we have adult and elderly human individuals that have latent TB infection, and then those with active TB disease. So this is essentially progression of TB disease. And here we're looking again, as I mentioned in the peripheral blood, we're looking at by ELISpot T-cell production of interferon gamma. And in tuberculosis, interferon gamma is a protective correlate. And so this is antigen specific. So these T-cells are responding to MTB antigens. And what you can see are that there are no real differences in the ability of these T-cells to respond between the elderly individuals and their adult counterparts. So they are responding in a functional manner. However, if we look a little bit further and start to phenotype these cells, looking here within, on the left, the CD4 compartment and the right, the CD8 compartment, you can see that particularly in the elderly individuals with increased disease progression, that there's higher levels of the exhaustion marker PD-1. And we also found that in the elderly individuals with active TB, that in their CD4 and CD8 T cells, they had decreased expression of a skin homing molecule called CLA. And so to us, this is suggesting that there are some age-related defects that are occurring in immune cells once they're exiting the blood or once they're in the tissue, and that these differences between peripheral responses and tissue level responses are resulting in the elderly being more susceptible when they're actually challenged in the tissue. And in this case, the tissue being the lung, as I mentioned, TB is primarily a pulmonary pathogen. So based off of this and some other data we have from the lab showing how aging impacts the lung environment and impacts susceptibility to infection, it leads us to this hypothesis that we'll test and I'll show you today. That is that with increased age, there are changes in the local tissue environment, and this impairs immune cell migration to and function in the tissue, which is resulting in decreased tissue immunity that we find in old age. And so I had mentioned the importance of the lung and how the elderly are more susceptible to respiratory infections. But we also know clinically that elderly are more susceptible to skin infections. And in this case, because I'm talking about TB, one of the ways to diagnose a previous TB infection is through using the delayed type hypersensitivity response, the tuberculin skin test, or TST. And in fact, we know that in elderly individuals, they had decreased or delayed tuberculin skin test responses. So again, a link between the skin responses and old age. And so this is actually the model that we're going to use, the tuberculin skin test. So I just want to briefly cover how this works. This is a really great model for us to probe in vivo cellular responses to vaccination and challenge. And so what happens in the TST, and many of you have probably received this in like a type of annual TB screening, is that just below the skin, tuberculin is injected, and this is a mix of different mycobacterial proteins. These proteins are picked up and processed by antigen-presenting cells, which then traffic to the lymph node. And in the presence of antigen-specific T cells, they're able to interact with their cognate antigen and the antigen-presenting cell. They traffic back to the skin, and you develop an inflammatory response about 48 to 72 hours later. And so this response, you can see here a positive test on an individual's arm. And I mentioned this can be due to TB infection, but it also can be due to BCG vaccination. So BCG, or Mycobacterium bovis BCG, is an attenuated vaccine that is the only vaccine approved for use in tuberculosis. And while it's not used here in the US, it is used worldwide in children. And so keeping this in mind, I'm going to talk about the design for this study. And so this was a small study that we did in coordination with the University of Oklahoma Health Science Center. And so our study utilized 4 male baboons. And in this case, we are defining the adult baboons as being under the age of 12 and the old baboons as being over the age of 16. And so to go over the study timeline, we started by vaccinating all 4 animals. So this vaccine was given intradermally on the upper arm with BCG at the standard human dose of 5 × 10⁵ colony forming units. And so we waited a few weeks for the animals to develop an immune response to the vaccine. And then we came back at 8 weeks. And at this point, we performed the tuberculin skin test. And so this is the skin challenge. And to give you an idea of what this looked like, these challenges were performed on the chest of the animals to give more space. And on one side, we performed the injection with tuberculin, and on the other side was saline to serve as the non-specific tissue control. And then I mentioned that it takes about 48 to 72 hours to develop the response to tuberculin. And so we waited 72 hours and came back and performed biopsies. And so all 4 animals had positive TST conversions at this point. And so we performed skin biopsies immediately surrounding the area of either tuberculin or saline injection. And so in the coming slides, I'll be talking about tissue homogenates that were obtained from these biopsies. And so again, saline will be serving as the control in the next couple of slides. So we first wanted to look within the skin tissue and evaluate measures of oxidation, because we know that with older age, there are higher levels of oxidation, and this can impact immune responses. And so we used electron paramagnetic resonance, or EPR, and we looked at levels of superoxide in the skin tissue. And so you can see here that in the skin tissue homogenates from old animals in response to tuberculin, that we did have higher levels of oxidation. And this is just shown here on the right in the spectra. We also wanted to look at levels of glutathione, which is an antioxidant. And similar to what I showed on the previous slide, there are higher levels of both reduced glutathione and total glutathione in the biopsies taken surrounding the site of tuberculin in our older animals. And so this may be increases in antioxidants in response to the oxidation. And in addition to the oxidation, we wanted to look at levels of immune proteins, so different proteins that are important in that immune response in the skin. And so we did this within the skin tissue homogenates by Luminex. And so here I'm showing 2 pro-inflammatory cytokines, so IL-6 and IL-1 beta. And you can see that the adult skin tissue in response to tuberculin, we have a very robust response of these pro-inflammatory cytokines, whereas the response in our older animals in response to tuberculin is decreased. We saw the same observation when we looked at the skin tissue and we looked at growth factors. So in this case, looking at GM-CSF and VEGF, you can see that the adult skin in response to tuberculin is responding significantly higher with the production of these growth factors. And then last, we looked at different chemokines present in the skin. And so here looking at CXCL10 and CCL20, which are important for recruiting T cells to the skin. And then CCL2, which we know is an important monocyte macrophage chemoattractant. And you'll notice that in the case of the adult skin in response to tuberculin, we're having a strong production of these chemokines, whereas the old skin in response to tuberculin, we're having a very blunted response. So having seen that the old skin, when challenged with tuberculin, had higher levels of oxidation and decreased levels of immune proteins that can help to generate this immune response, we wanted to ask, does this impact the ability of cells to migrate to the tissue? And so in order to test this question, we used an in vitro PBMC migration assay. So peripheral blood mononuclear cells, or PBMCs, were obtained from the blood. They were added into the top chamber of the assay. And then tissue homogenates were, from the biopsies, were added into the bottom chamber, and the PBMCs were allowed to migrate. And so we tested this in 2 different setups, the first being what I'll call the homogeneous migration assay. So this is using the adult PBMCs in response to adult tissue. So either biopsies taken from tuberculin or saline biopsies, and then the old PBMCs in response to old tissue. And so having seen that the old tissue had blunted immune responses, we were expecting to see that this would impact the ability of PBMCs to migrate. However, we actually saw the opposite, where PBMCs from our older BCG-vaccinated baboons actually had increased migration. And so we wanted to ask, is this due to this homogeneous setup that we have? Is it that the old animals, the old PBMCs are responding due to, say, something in the old tissue? And so we actually wanted to do the reverse of this. And so here, setting up what we're calling the heterogeneous migration setup, where essentially you're taking adult PBMCs, migrating in response to old tissue, and vice versa. And we see the same phenomenon here, where really, regardless of the source of the tissue, old PBMCs are migrating more. And so since we find that PBMC migration is functional in our older vaccinated animals, despite the oxidation and immune protein changes in the skin, we wanted to ask, are these cells functionally altered? And so to test their ability to function, we wanted to look at their ability to produce cytokines in response to antigen stimulation. And so here we took PBMCs from our adult and old BCG vaccinated baboons, and we stimulated them for 5 days with CFP, which is a mix of mycobacterial proteins. And by Luminex, we looked at the ability of the PBMCs to produce different cytokines. So here, just to orient you, we're looking at a couple of time points after tuberculin skin test was given. and we're looking at the ability of these PBMCs to produce IL-1 beta. And we find that there's no impairment between our age groups. We saw the same thing when looking at other pro-inflammatory cytokines, here showing TNF and IL-6. And then we also wanted to look at IL-2 and interferon gamma, which are important T cell cytokines. Again, we find that our old PBMCs are functional in their ability to produce these cytokines in response to antigen stimulation. So we thought, well, maybe then the PBMCs are phenotypically different, which is resulting in the changes that we saw in the migration. And so we wanted to phenotype them using flow cytometry. And for the sake of today and limitations in time, I'm just going to summarize some of the changes that we saw in our flow cytometry experiments. But overall, we saw that there were several age-dependent changes in our PBMC populations, but these changes were present prior to the vaccine being given. So this was present at day 0 before animals received BCG. And so just to summarize some of these age-associated changes in PBMC phenotypes, we found that with age, there was a decrease in the lymphoid to myeloid ratio. We also saw increases in what we're defining as natural killer and NKT cells and gamma delta T cells. And then we also saw a shift in our CD4 T cell compartment to a higher percentage of central memory cells. And it's important to note that these changes have been described in aging literature using different model systems. So this wasn't surprising to us. It recapitulates what you see in different models. And importantly, we saw it before the vaccine. So this was really age dependent and not due to BCG. And so the last question we wanted to ask was whether or not perhaps there were changes in chemokine receptor expression on PBMCs, that these changes could be driving what we see with old PBMCs having higher levels of migration. And so again, we looked at this by flow, and I will just summarize this here for sake of time. But we found that within the T cell compartment of old animals, looking at both CD4 and CD8 T cells from BCG-Vaccinated, that they had higher levels of CCR4, which is an important skin homing receptor. And although these increases were not significant, they did trend higher. And so it could be one of the pieces playing a role in potentially why we're seeing higher levels of in vitro migration in our older PBMCs. So to conclude what I showed you today, we have this project where we've established a baboon model that allows us to study BCG vaccine-induced tissue immunity. And so within the skin, we perform a tuberculin skin test, and we're using aged and adult BCG-vaccinated animals. We found in vivo, so within the biopsies of older animals, higher levels of oxidation and decreased levels of immune proteins. However, when we look in the peripheral blood, we find in older animals, PBMCs, at least in vitro, are functionally very similar to their adult counterparts when we're looking at their antigen-specific responses. And we find that they have increases in cell migration. And so we're thinking that there are age-related changes that are occurring in the tissue that are impacting cellular functions in vivo. And so this is kind of a question mark of kind of where we're going next. And we're thinking that potentially structural changes in the skin may be impacting the ability of these cells to migrate in vivo. And so we have a couple of studies that will be coming up. They were on hold due to the pandemic, but we'll be able to investigate this in a larger sample size. And so with that, I will thank my lab at Texas Biomed and my mentor, Joanne Turner. And I also want to thank those that we worked with at Oklahoma, and then acknowledge the funding, the T32 training grant. And I can take any questions now. I think I saw something in the chat. Okay, let's see. In migration assays, is there any evidence that different PBMC types perform better or worse than others? So, and then this from Adam as well. Have you looked at any potential differences in senescent cells among groups in your PBMCs? So in terms of the migration assays, I haven't been able to, just due to the number of cells that migrate and being able to try to get them from the bottom of the chamber, it's just been technically challenging to try to phenotype those that do migrate, which is why we went the route of trying to look at potentially receptor expression, because we know that receptor expression can help give us an idea of what would be migrating. But there's definitely the possibility that that there are differences in how certain cells are migrating. I can tell you that overall, when we look phenotypically at the populations, that they are overall pretty similar between the age groups, and they aren't impacted by the vaccine. And so it is less likely, I would say, that there's a major difference in phenotypes that's driving the migration. But at this point, I can't say for sure. And then looking at senescent cells, I have not looked at any senescent markers. I'm instead going to be focusing on looking at senescent markers in the tissue, because we do think that it's less changes in the periphery and more changes in the tissue that are what are impacting the responses in vivo. And so we have some, we're collaborating to do some tissue imaging, and that will give us a better idea of changes in the tissue that may be resulting in some of the findings in vivo. And then what animal are you using? So these are baboons, and yeah, so these were baboons. They were a mix of the Hamadryas and the olive baboon. These were through Oklahoma at their primate center. But in follow-up studies, we're going to be using olive baboons, and then we're also going to be using rhesus macaques. The assay, could it be done in marmosets? Certainly, we just haven't decided to do any work in marmosets. We've been trying to use we've been using primarily baboons that is the study that we have set up for Texas Biomed, just because they do have higher sequence homology to humans. But certainly, I mean, marmosets would be easier to work with and cheaper. And then, does this animal naturally?
We're going to move on, and if we have time at the end, I'm sorry, we can answer any final questions.
There's only one left, so we're fine.
Okay.
Okay. So can you see my screen now?
I can see and hear you, so we're doing well.
Perfect. Okay. So hello, everyone, and thank you for joining this Barshop seminar. I'm going to talk about Tau-mediated nuclear envelope invagination causes RNA accumulation. Okay, so lamin dysfunction mediates neurodegeneration in tauopathies. Lamin protein is a nuclear protein, and as you can see in this schematic, it is associated with the chromatin as well as cytoskeleton through a complex called the link complex, that these are the component of that components. So lamin protein, it provides physical strength to the nucleus, and also it gives the genome and architecture. It has been shown in many cancers and also in progeria syndrome that the dysfunction in lamin protein is associated with cell death and toxicity observed in these diseases. In 2016, Frost et al. Also showed that disruption of the lamin nucleoskeleton drives neuronal death in tauopathy. So I'm showing a picture from the paper showing that the Alzheimer's disease patients, they have this nuclei with dysfunctional lamin protein, and you can see that the nuclear envelope is invaginating and it's unfolding, but we don't have this phenomena in the control. And also, when they stain for the nuclear pores, it shows that there is this co-localization of nuclear pores within this invagination, which shows that the property of the nuclear envelope is coming inside to the nucleus via these invaginations. So, and I think everybody knows, but talking about Alzheimer's disease and the tauopathies in general is that basically we have this pathology, we have this aggregation of the phospho-tau in these neurons that cause this toxicity. So showing more of the results from the paper Frost et al. In 2016. They measured this invagination and they observed that there is a significant increase in the nuclear invagination in Alzheimer's disease patients versus the control. And when they performed the same analysis in Drosophila model of tauopathy, they observed that nuclear invagination is again increased in these models. In 2018, Cornelison et al. They decided to see what is inside this invagination. They performed this fish IF experiment using poly-D-T probe, which is binding to the polyadenylated RNA, and they showed that inside this invagination there is this accumulation of the RNA, mRNA, that you can see in the picture here, and they might come in different shapes. They might like kind of engulfing the nucleus or be inside the invagination. So by genetically manipulating the RNA export system, they observed that there would be less toxicity associated with these Drosophila models, and also there are less nuclear envelope invagination. And these data suggest that nuclear envelope invagination promote neurotoxicity by increasing RNA export. More pictures showing that there is this specificity for the poly-D-T probe, that when they use it in the tau and the control Drosophila, you can see that the probe is specific to the mRNA, because in the presence of poly-DA, there is no staining. The same results was showing for human Alzheimer's disease, showing that inside these invaginations, we have poly-D-T. And when I joined the lab, we decided to further investigate nuclear invagination and nuclear accumulation. So, and using a new model, I decided to use induced pluripotent stem cell derived neurons. And before I show the results for nuclear invagination inside in these models, I'm just going to show some of the characteristics of the neurons that I'm using, and to show that at the time of the experiments, these neurons were completely mature. So here are the pictures showing using a neuronal markers MUN, which is a nuclear neuronal marker, and MAP2, showing the maturation of these neurons, and also these neurons are expressing phospho-tau as well as GFAP. So GFAP is a marker of glial cells, and this shows that we have a combination of the glial and neuronal cells in our culture. More images showing the characteristic of these neurons in vitro. And when I did the fish IF experiment to show that whether we have RNA, polyadenylated RNA within this invagination, we observed that yes, the presence of these polyadenylated RNA inside invaginations in these iPSC-derived neurons, which have the MAPT mutations. More images of that, and this was an important experiment because now it is showing that we are observing nuclear invagination and observing the presence of polyadenylated RNA inside the invagination among different models. So we observed it in Drosophila model of tauopathy, in Alzheimer's disease patients, and also in iPSC-derived neurons. The second thing that I did with iPSC-derived neuron was to create spheroids out of them. And spheroids are interesting in different ways because, first of all, this is the way that we make the spheroids. So out of the induced pluripotent stem cell, we just put them in culture, and after 50 days, they form these ball shape that are completely mature. So they are electrophysiologically mature. They form functional synapses, and they have similar transcription profile to human fetal brain. And size-wise, they are the size of a mouse brain at 12.5 weeks. So at day 61, they are very similar to the mouse brain. Some of the characteristics of these spheroids is that the mature human cortical spheroids, they have diversity of neuronal and glial cell population, which makes them which actually makes them to better reproduce the in vivo physiology of the brain tissue. In many drug delivery experiments, they observed that human cortical spheroids, they provide more human relevant data compared to the animal models, and this might be because of interspecies differences. And also they are interesting because they can be used for large-scale drug screening in vitro. And they are interesting in a way that we can use them for personalized medicine. So the iPSCs can come from unique patient populations and then can create mini brain out of their cells. This is the way that I make these spheroids in culture. So I just put them in 96 well plates and just observe their formation through their maturation. So in the early days, around 25 days, we are still able to see neuronal rosette at the periphery, but as we go further, it's like we just see a ball of cells. So it's not we are not able to observe any structure with the light microscope at this point. And then here I'm just showing that there are different markers and growth factors that needed to be added at different time points to get to the mature human cortical spheroids. On day 38 of my culture, I take some of these spheroids out and sectioned them and decided to check for the progenitor neuronal markers to see if they are on the right track. So we stained for nestin, PAC6, and almost all of the cells inside the spheroids, they are expressing these signals, these markers. This is a higher magnification showing that all of these neurons have the progenitor markers. And on day 50, when we stained for neuronal markers for the tau here and also for GFAP, again, we saw that the positive staining in these spheroids showing that they are completely mature. The NEUN and MAP2 staining shows the maturity again, and the most important thing for us is the Tau5 or the phospho-tau expression that is associated with the toxicity observed in tauopathies. So not only they are expressing the phospho-tau, they are also showing this increased level of nuclear invagination that might be because of the presence of the phospho-tau or maybe like it's still I still need to figure this out, or maybe if the cells are very close to each other. So anyway, in general, it makes them these spheroids and also the 2D culture of iPSC drive neurons a very interesting model to further investigate the impact of RNA accumulation within invaginations. So one question that I was trying to answer during my research was that other than the polyadenylated RNA, what type of RNA are inside these invaginations? And I was thinking that circular RNA might be present inside because of some of their specific characteristics that I'm going to explain later. First, I'm just going to talk about how they are generating in the cells. So circular RNAs, they form during a back splicing event when an upstream splicing acceptor a donor is binding to a downstream splicing acceptor and forming these circular RNAs. And they can be in different shape and size. They can either be in 1 exon, 2 exon, or they can have intron in between. They are very interesting because circular RNAs, they are specifically expressed in the human brain, and they are upregulated during neurogenesis and accumulate during aging. Despite lack of polyadenylation and capping, they are able to localize in the cytoplasm, and they are very stable compared to the linear mRNA. The circular RNA, they can shape the gene transcription, because they can form these sponges that microRNAs are able to bind to them, and then they form the sponge and then trap them and won't let them to function. Also, they can compete with linear RNA during transcription. Another important thing about their formation is that the presence of transposons and microsatellites in the flanking regions of the circular RNA facilitate their formation. So all of these make circular RNA a nice target to study. So when we, in our Drosophila model of tauopathy, when Paul Ramirez from our lab, he did the bioinformatics on these RNA libraries in the Tau and control, and we find that there is this increased level of circular RNA in the Tau compared to the control. And it was not that there is only an increase in the circular RNA level. There are certain circular RNAs that appear only in the Tau, and they are absent in the control. So when I did the fish staining for the circular MBL in the control and Tau, we observed again the increased level of intensity for the circular MBL in the Tau compared to the control. Doing the dPCR to check for the expression level of circular MBL, we saw that circular MBL is like highly increased in the Tau compared to the control. And this experiment was necessary because the functionality of circular MBL need to be validated. If, for example, the linear MBL is also highly expressed, there might be questions that maybe the circular MBL is coming because of the more transcription that is happening for that gene. But now that we have this increase in only the circular MBL, it means that there are functionality, that there are functions associated with this increase. And lastly, when Elias Gonzalez from our lab, he did the tunnel assay and the Tau and the MBL knockdown plus Tau flies, we observed less toxicity, which means that this increase in the level of expression of the MBL is associated with toxicity in the Tau flies. So the experiments for the circular RNA are still ongoing, and the most important question is to show whether these circular MBL are located in the nuclear invaginations or not. And the next thing that I'm planning to do is to see what are the major bindings to the circular MBL. If we have any type of RNA or protein that are kind of trapped in the invagination because of the presence of circular MBL. So these are the experiments that I'm currently doing and working on. Just to summarize what I talked about today, I talked about nuclear invagination that is happening among different tauopathy models, Drosophila, iPSC-derived neurons and spheroids, and also human Alzheimer's disease. Also showed that polyadenylated RNA accumulates in nuclear invagination in all tauopathy models, and circular RNA biogenesis increase in Drosophila model of tauopathy. And also the human cortical spheroids are physiologically relevant model with more faithful recapitulation of brain spatial organization. With that, I want to thank all of the members of Frost Lab, my T32 Biology of Aging grant, and the R01 for Dr. Frost. Thank you.
Thank you so much.
So there is a question in chat asking, Gary Scott asking, did you test any SIRTs, deacetylation, capable of reducing tau phosphorylation? No. I mean, in what? In the spheroids, you mean? In the iPSCs or in the flies? I didn't get your question.
Hello, can you hear me? This is JP. I'm very excited to hear about your 3D cultures. Very cool stuff. I just wondered, do you see tangle-like structures? I think you put some Tau histology, Tau staining, but I couldn't really appreciate. Just wondering, and you showed the data from these Tau mutants. I wonder if you have just Tau wild type controls and whether you see the invaginations there or not. Obviously, I guess you would expect to see much less.
Yes, so the expectation is to see much less, and I am working to get those spheroids, but at the moment I don't have them. So hopefully for the next presentation I will have the results from the control as well. So the phospho-tau staining is this is the phospho-tau staining. Oh, here. This is the phospho-tau staining in the spheroid. Yeah.
So it looks like not too much of tangle-like, right? What do you think? Like strong body accumulation, rather than more axonal or processes.
So one thing that we might observe like better for the phospho-tau is that the maturation date, this is the minimum that I kept them in culture. Sometimes people keep them for 120 days to have the full expression of the phospho-tau. So I think this is the beginning of the expression of the phospho-tau at day 50, and usually like it is normal to keep them till day 70 in culture. So there should be, I think I need to do like a timeline, do different, for different days, just do the Tau staining, phospho-tau, to see how the expression is.
Yeah, makes sense. That's exactly what I was thinking, like whether you see it or not now, that, waiting longer, probably you eventually will see them. And I was wondering about how long you would need to wait. But yeah, it's very cool stuff. And you're eventually gonna know that.
Yes.
Hearing more about this, yeah. Thanks.
So there is another question in the chat from Dr. Musi asking why the polyadenylated RNA accumulates in the invaginations. So there is this hypothesis that there might be the excess expression of the RNA in these flies and in the tauopathies. We could not confirm it yet, but if there is like this increase in the level of transcription, then some of these RNAs are trying to get out of the nucleus, and they just find invagination, because the idea is that the nuclear invagination are bringing the function of the nucleus inside. So whatever the function for the nuclear periphery we have now, these invaginations are doing that, and they, like if we have excess level of RNA, then they just trap inside and they cannot get out.
Well, if there are no final questions, I want to say congratulations to all of you on the great work you are doing. You all have a lot of data, which speaks to the hard work you're putting into it. And also want to say just an appreciation to all the mentors that are assisting with these projects. So we appreciate everyone attending, and we will see you next week.